Hello, everybody. Welcome back to Genetics. We are going to be shifting gears now from DNA replication. We're going to be moving on to something a little bit more recent in our understanding, and we're going to go to DNA manipulation techniques, things used in genomics and genetic engineering, and just basically molecular biology lab techniques that are useful. So what are we going to be learning about today? Well, we're going to learn about a couple of specific methods of DNA manipulation. At least three, possibly four, if we can fit in the Sanger sequencing. Two of these are going to be important for upcoming labs in the next few weeks, restriction enzymes and polymerase chain reaction. And you may have encountered some of these in previous bio classes. You might have even done some restriction digests and PCR in the past, but I'm not sure. So with that, we're going to be learning about three or four different techniques today. The first one we're going to be talking about is really key basic information that makes everything else possible. And that's denaturing and annealing. So what are denaturing and annealing? Well, quite plainly, denaturing is separating the two strands of a double helix from each other, and annealing is bringing them back together again. That is the difference between them at its simplest. The two strands of the double helix, as we've discussed many times, are held together by the hydrogen bonds between base pairs. And the nucleotides in a single strand of DNA are held together by covalent bonds along the phosphate backbone. You know, the phosphate of one binds to the sugar of another, which has a phosphate attached to it, which binds to the sugar of another, and so on. And those are covalent bonds. And those covalent bonds are significantly stronger than the hydrogen bonds holding the base pairs on opposite strands together. And that's how a helicase is able to separate the two strands of DNA just by wedging itself in between them, because it can, hydrogen bonds are only really strong enough that if they're in just the right proximity, they will hold, but if the atoms are pulled apart even a little bit, then the hydrogen bond just breaks. I mean, really all it is shared electrons, and those electrons just stop getting shared. So if you were to take a double helix of DNA, and if you had really microscopic tweezers, and you grabbed one strand at a base with one set of tweezers, and you grabbed the opposite strand at the same base level with a different pair of tweezers, and you pulled them apart, you could break that hydrogen bond very easily. Now, what else breaks hydrogen bonds? Well, honestly, heat. Heat breaks hydrogen bonds very easily. Because what is heat but just increased energy in atoms? And the increased energy makes the atoms shift and vibrate and migrate, with increasing quantities and increasing distances. And once it gets hot enough, about 90 to 94 degrees Celsius, the hydrogen bonds between any given pair of bases will break. And they will, separate from each other. The two bases on opposite strands will separate from each other. Well, if you do that to a long double helix that's, 1,000 bases long, just to throw out a number, all throughout that 1,000 bases, there's going to be individual base pairs separating from each other. And when all those base pairs separate, then there's nothing holding the two antiparallel strands of DNA together. And so they just sort of drift apart. So as long as they're retained at that heat, they will drift apart. Now, I know I've said in the past that DNA does not like to be single-stranded, and that is true. But at this high temperature, there's really nothing to be done for it. The DNA just floats around as a very thermally active single strand. Now, once that temperature starts to drop and the solution that the DNA is in starts to cool, then conditions become energetically favorable for base pairs to start binding again. And one of the tricks about it is the slower the cooling is, the more structured the binding will be. And the faster that cooling is, the more haphazard the binding will be. And let me explain what I mean here. So let's say that we have a little Eppendorf tube with a solution of salt water with DNA in it. And we put that in a water bath that's heated up to 94 degrees. The DNA in there, within a minute or so, will be completely single-stranded. And all the individual chunks of DNA will be floating around as single-stranded DNA. If we take that 1.5 mil Eppendorf tube out of the water bath and we just set it on the counter, room temperature, what's going to happen? Well, the temperature of the solution is going to slowly start coming back down to room temperature. And the DNA in there is going to become less and less energetic. And while that's happening, the various different strands of DNA are going to start bumping into each other. And an A is going to find a T and it's going to bind. And a G is going to find a C and it's going to bind. The trick is, any old A is going to bind to any old T at first. It doesn't necessarily mean that this A is going to bind to the correct T on the opposite strand that matches where it should be. It's just going to find any old T and bind to it. But if the bases on either side of that A can't also bind to the bases on the other side of that T, well then it's not going to be a stable binding. You're not going to start zipping up the double helix because you've just got one base matching and the surrounding bases don't. So the sort of inability of the neighboring bases to bind is going to put strain on the helix that's going to essentially break that one single set of hydrogen bonds. And so it's not going to stay long as a stable base pairing. But the A is then going to find a different T to bind to. And the same thing is going to happen over and over until it hits a T where the neighboring bases can also bind. And then can the bases neighboring those also bind what's on the opposite strand? And eventually, the bases are going to find their perfect match in the opposite strand, and then the whole strand is going to zip up. And you're going to get your double helix back beautifully the way it was before, no problem, no change. That's what happens when you cool the DNA slowly. That's annealing. What happens if we cool the DNA quickly? Well, so let's go back to that Eppendorf tube full of DNA. And this time, instead of taking it out of the hot water bath and putting it on the counter, we take it out of the hot water bath and we shove it in a bucket of ice. And now the temperature is going to drop much more rapidly inside that test tube. And the DNA is going to cool. And this is going to be like a game of musical chairs. Everyone's going to be running around at high speed. You're going to be, struggling and jumping over each other to get to a match. And it's not all going to work out well. Those first matches that the A and the T were trying to make in the slow cooling, those are not going to be broken this time. The random strands of DNA are going to make random bindings. It could be to, for instance, if we take a random A, it's going to bind to a T. That T could be on the same strand of DNA as the A, just 50 bases downstream or 1,000 bases downstream, what have you. Or it could be on a different strand of DNA, but not the correct different strand. Or it could be on it could be anywhere. And you're going to wind up with a whole lot of random base matches surrounded by a whole lot of non-matches. And the whole thing is going to be disjointed and confused. There's going to be incorrect bases all over the place. There's going to be loops, and strands of DNA are going to fold back on itself and bind to itself. It's just going to be a nightmare. That's what happens when you rapidly cool the DNA. So, scientists take advantage of this in so many different ways. And the first, most obvious way is, let's say that you have a chromosome. And that chromosome is, 150 million bases long. It's going to take a very long time for a denatured chromosome to anneal completely. It's going to take a long time, because that's a lot of bases of DNA to correctly line up and bind. But what if, in the test tube with this chromosome, we throw in a bunch of, 20 base pair long sections of DNA, or 1,000 base pair long sections of DNA? I mean, compared to 150 million, 1,000 is nothing. It's tiny. And if those 20 base pair or 1,000 base pair or whatever small sections of DNA perfectly match a part of the chromosome, then as the chromosome is going through this slow cooling period from denatured and trying to anneal, what's going to happen is the small fragments of DNA are going to find their matches on the chromosome very quickly compared to the rest of the chromosome, and they're going to bind. And then as the rest of the chromosome starts lining up to itself and binding, all the rest of the 150 million bases are going to line up beautifully, except there's going to be this 1,000 base stretch where there's already a double helix there, because this other thing bound in its place. And so the chromosome, the one strand of the chromosome is going to be bound to this 1,000 base long stretch. The other strand of the chromosome is going to be bound on one side of that 1,000 and bound on the other side of that 1,000, but it's going to have 1,000 bases just sort of sticking out into the breeze. And we can take advantage of that too. So for instance, if instead of just throwing in a single-stranded 1,000 base thing, if we threw in a double-stranded 1,000 base thing, then you have a good chance of each strand of your 1,000 bases binding to one side of the chromosome and the other side of the chromosome. And so then the chromosome, when it reanneals, is going to be this huge stretch of double-stranded DNA with, somewhere in the middle of it, a 1,000 base pair stretch where you have two parallel double helices that end up next to each other, forming a single double helix at either end. And if we, instead of just throwing in random 1,000 base pair DNA, we can throw in modified DNA, where the phosphate backbone actually has other chemicals attached to it, or other molecules attached to it, I should say. And in this case, we refer to these small regions of DNA that we are inserting as either primers or oligonucleotides. That's the technical name is oligonucleotides. They're generally referred to as primers. And in this case, think back to transcription replication that we talked about in the previous lecture, where you needed an RNA primer for topoisomerase to work correctly. This is the same idea. It's just a short stretch of DNA that binds to a larger DNA, and it's used to prime a reaction. In this case, not necessarily a replication reaction, but a reaction of some kind. So let's look at this in action. I've mentioned the protocol FISH in a few previous lectures. And to remind you, this stands for fluorescence in situ hybridization. In situ meaning in place, hybridization, we are hybridizing DNA, and we are making it fluorescent. Fluorescence in situ hybridization. So in this case, we're going to take a 1,000 base long double helix of DNA, and we have bound to the phosphate backbone of this DNA some molecules that give off fluorescence when you shine UV light on them. Okay? And that's all it is. There are multiple different molecules that biologists have identified and use constantly that will fluoresce under UV light. This is how bioluminescence works. If you're familiar with GFP, the green fluorescence protein, or any of its variations, red, yellow, cyan, all the various different fluorescence proteins, jellyfish, deep sea organisms, this is how they fluoresce. So we're going to take this 1,000 base pair double-stranded DNA that is bound to this fluorescent molecule, and we're going to throw it into a test tube with some chromosomes. And we know that the 1,000 base pairs binds to one of the chromosomes somewhere. It is the sequence of a gene that we have identified, and we want to know where on which chromosome it is. So we take that test tube and we heat it up to denature all of the DNA, and then we let it anneal slowly. And just like I said on the previous slide, one strand of that 1,000 bases is going to bind to one strand of the chromosome, and the other strand of that 1,000 bases, the other strand of that short double helix, is going to bind to the corresponding opposite strand of the chromosome. And as you see in the bottom center of this image, the chromosome is double helix all the way around on either side, but for this one short stretch, the 1,000 bases we put in is blocking the chromosomal double helix from forming, and instead it forms two hybrid double helices, where one strand is chromosomal and the other strand is our primer. Okay, well, great, we've got this binding, what do we do with it? Well, in the case of fish, what's typically done is that DNA is then put on a microscope slide, it's put in a microscope, and UV light is shown on it, and you get a karyotype where part of one of the chromosomes fluoresces and shines a bright fluorescent red or green or whatever. And that's the X-shaped sister chromatids on the left side on the bottom where it says analyze. This can be done not just with chromosomal DNA and karyotypes. This can be done at much smaller scales and finer levels. So, molecules that show a location of something, like a fluorescence molecule that shows where things are located on a chromosome. These molecules are called reporters because they report to the scientist the position that they are at, or they report to the scientist that an experiment is going correctly or incorrectly. They provide the scientist with information. There are other molecules that we refer to as quenchers. And in this case, quenchers are things that prevent the reporter from reporting. Strictly speaking, in terms of fluorescence, when you quench fluorescence, it just means that the photons that are being used to activate the molecule to glow a fluorescent color, that photon is being absorbed by the quencher and nothing will fluoresce. That's the technical explanation. You don't necessarily need to understand that. Just know that quenchers work when they are in close proximity or even connected to reporters, in this case. And they prevent the reporter from reporting. It's like an older brother standing over you holding your mouth shut so that no one can hear you. There's literally thousands of uses for this type of a system. There are two of these uses illustrated on the slide that we're looking at now. So the one on the left is a way of telling whether a specific region of DNA has been replicated or polymerized. So if we take a primer, in the image it's labeled as a probe. In this case it could be either because we are probing the system for information, but we are doing so with an oligonucleotide primer. So either is correct. If we have this probe or primer, a small stretch of DNA that we have generated in a lab, and we put attached to it on one part of it a reporter and on the other part of it a quencher, then nothing's really going to happen. Any kind of fluorescence that the reporter might have is going to be stopped, quenched, hidden, whatever you want to call it, by the quencher. Now, if we want to find out if the region of DNA that matches the probe sequence, if we want to find out whether or not that sequence has been replicated or polymerized, then we throw that into a cell or into a bacterium or even into a test tube that has all the necessary components for replication, and we initiate replication. And what will happen is, as the polymerase, as Pol3 comes cycling along down the double helix to polymerize the bases in front of it and extend the double helix, as it hits the probe sequence, it's going to kick it off of the DNA. And if we're using Pol-1, which I mentioned has exonuclease activity in the 5' to 3' direction, then it's going to remove the bases that are in front of it in its way one at a time as it progresses forward one at a time. And that's what we're seeing with strand displacement and cleavage on the left side here. And if the topo-1 topoisomerase cuts the reporter off of the primer, the reporter starts to float away. And now it's no longer in close proximity to the quencher. And all of a sudden the reporter is now shining fluorescent. And so we now know that the polymerase has reached the section of DNA that we were interested in. This is also incredibly frequently used with looking at locations of RNA. And that is a technique called in situ hybridization, not to be confused with fluorescence in situ hybridization. They are incredibly similar techniques used for slightly different purposes. But when you look at the location of RNA in a cell or in an organism, that's a great way of finding out where a particular gene is active and being expressed. And so that's a great way of trying to understand the function of a given gene. And we use fluorescent reporters in that kind of situation. In a fluorescent reporter attached to a section of DNA that anneals to a single-stranded RNA or DNA. So all of these are just ways to describe to you that denaturing and annealing DNA can lead to dozens of possibilities of finding out information. Just the very simple act of forming and breaking hydrogen bonds. We can do so much work. But that's not all we can do. So let's talk about restriction enzymes. Technically speaking, they're called restriction endonucleases because they are cutting nuclease DNA. They are breaking the phosphate backbones of DNA. So what are restriction enzymes? What do? They are proteins. They are proteins that bind to specific sequences of DNA. And if you remember the lectures on the structure of DNA, that means that when they bind to the DNA, they are reading the shape of the major groove. And when they see a specific three-dimensional structure of the major groove, they can bind to it like a 3D jigsaw puzzle piece. And if it's the wrong series of bases, then it's not the right shape and they can't bind to it anymore. And most restriction enzymes are actually symmetrical in that they can bind regardless of the direction of the sequence. And that means we have palindromic DNA sequences. And I don't mean palindrome in the way of mom, dad, race car, or Madam, I'm Adam, where it's all the same letters forward to backward. The DNA version of that is it's the same letters from 5' to 3' regardless of which DNA strand. One way to think of it is you're not doing a horizontal mirror image of the letters, you're doing a 180-degree rotation. So the first example I give here, ATGCAT, if you think through what is the complementary sequence to that, reading from 5' to 3', the complementary sequence is ATGCAT, which is the same sequence. So many restriction enzymes bind to these palindromic sequences. Not all of them, but the vast majority. Some of them bind to a sequence of 6 bases. Some of them bind to a sequence of 4 or 8 or 16 or what have you. There's one that I think binds a sequence of 75 bases. But really it binds 6 or 8 bases here, and then 70 bases downstream from that, you have to also have a specific 6 or 8 bases. And as long as it can bind those two separate things a specific distance apart, then it'll cut the DNA. So why do restriction enzymes exist? What are they? They are a part of the bacterial immune system. Now, as I'm recording this on Wednesday the 7th, Jennifer Doudna and a few others, it was announced, received the Nobel Prize in Chemistry for the, not so much discovery of, but modification and utilization of CRISPRs for DNA modification. CRISPRs are a form of, I believe, yeast, possibly bacterial immune system. Now, restriction enzymes and CRISPRs are different. Both of them cut DNA, but through different mechanisms. The reason that bacteria have restriction enzymes is they're fighting infection every bit as much as we are. And if you remember back to the Hershey-Chase experiment, the infection that bacteria have to fight is phage, or bacteriophage. And these are viruses that attack bacteria. They land on the bacteria, they inject their RNA or DNA into the bacterial cytoplasm, and then that DNA gets copied and gets used to make more viruses that eventually kill the bacterium. So obviously the bacteria do not like that, and they want to prevent that from happening. So they look for double-stranded DNA that's floating free in the cell, and they chop it up. And so they have evolved numerous different restriction enzymes that will match numerous different sequences. Each enzyme essentially matches one sequence. There's some slight variation to that, which I'll get into in a little while. Or actually, I get into it in the lab for this week. But how does the bacteria not chop up its own DNA? Because, I mean, if you look at that 6-base strand, you know, what are the chances that a bacterial chromosome that's 20 or 40 kb long is going to have A, T, G, C, A, T all in a row? Well, it probably will, a couple of times. So how does the bacteria prevent itself from digesting its own DNA? And in this case, what the bacteria does is it has other proteins called methyltransferases that attach methyl groups to the A or the C of its various DNA sequences that prevent the restriction enzymes from recognizing it, because adding a methyl group changes the three-dimensional jigsaw puzzle piece shape. And so the enzyme can no longer recognize that. The base can still function as a nucleotide beautifully. It still binds to the opposite base pair, and it can still get replicated and translated, or transcribed rather, but it can't be read by a restriction enzyme. So restriction enzymes were first discovered in the 1960s, and it slowly began dawning on scientists that they could be used by scientists to work our will on DNA. And this is an interesting anecdote that I've mentioned Cold Spring Harbor Laboratory a couple of times. It's a private research lab on Long Island. And there's been a lot of tremendously monumental, formative molecular biology research done there over the past 80 years, let's say. Jim Watson of Watson and Crick fame and recent racist infamy used to be the president of Cold Spring Harbor Labs. And he presided over it from the late 1960s to the mid-1990s. And at some point in the 1970s, one of the scientists working there, Richard Roberts, began purifying restriction enzymes from bacteria. And now, first off, what do I mean by purifying? Well, he would take a culture of bacteria that had millions and millions of bacterial cells in it, all of them identical, and he would break open all of the bacterial cells through a chemical process and extract all of the protein from it and get rid of the DNA and the cellular debris. And then by having all of that protein, he could then treat it with various different concentrations of salts and pH and temperature to get rid of certain types of proteins. And he would keep doing that until he wound up with a pure solution of just one protein type, and millions of copies of that one protein, but just the one protein type, and it would be still functional. So in the case of a restriction enzyme, he would have a test tube full of millions of copies of restriction enzyme, and if he added some DNA to that, then the restriction enzyme could chop up that DNA. So that's a purified enzyme in this case. And he spent a chunk of his time purifying these enzymes. While he was doing that, he also happened to, through other experiments he was doing, discover the process of mRNA splicing and win a Nobel Prize. So that raised his stature and his cachet at Cold Spring Harbor Labs. And as he was purifying all these enzymes, some of the other scientists at the labs started realizing, well, hey, I could cut my DNA that I'm using in my lab with these enzymes, and I can do all kinds of new, neat things with it. So, hey, Rich, can you lend me a small quantity of that, please? And, okay, sure, here, have some. And then other scientists at the lab would say, oh, I could use some too, because I got this really... Okay, fine, have it. And then word got out, and scientists at other laboratories, not at Cold Spring Harbor, like at NYU and Rockefeller, and up in Boston at Harvard and Woods Hole, Massachusetts, and Brookhaven National Labs, all over the place, people started saying, oh, hey, Rich Roberts over at Cold Spring Harbor has these really awesome, pure, and highly functional restriction enzymes that you can do so many amazing things to your DNA with. You can cut the DNA and attach it back together again differently. So everyone started writing to him saying, can you please send me? And then he would say, okay, fine. Until one day, finally, he went to Jim Watson and he said, listen, one Nobel laureate to another. We can't keep doing this. I'm spending all of my time making more protein for all of these other people, and it's taking all of my grant money, it's taking all of my time, it's taking all of my resources, and I'm getting nothing in return, and I can't do my own research anymore. It's just in the way. So I have an idea. Let's be entrepreneurs here, and let's form a company whose sole purpose is to sell people purified restriction enzymes. And Jim Watson basically laughed in his face and said, no. The pursuit of science is pure and cannot be adulterated by the pursuit of money as well. Cold Spring Harbor Laboratory will never profit off of our research. We do it solely for glory or what have you, or the benefit of mankind. And Rich Roberts said, all right, fine, I'm out of here. And he co-founded a company called New England Biolabs. And it's referred to as NEB for short, and it is one of the foremost suppliers of restriction enzymes and other purified proteins to molecular biologists around the world. And I gotta tell you, that company did really well, and is still doing very well. And Rich Roberts has not regretted his decision to found it for a single day of his very charmed life. So let's get back to restriction enzymes and get off of that particular tangent. When I say that they cut the DNA, what do I actually mean by that? Well, they physically grab the DNA and they break the covalent bonds of the phosphate backbone. They just destroy the bond connecting the phosphate of one nucleotide with the sugar backbone with the next nucleotide over. And they do this, most of them do it on both strands of DNA. And there's two ways that they can do that. One is they look at a specific pairing of bases and they cut right next to that pair, so that you wind up with two double helices that both end, boom, at a dead end. And it's double helix all the way up to the end. That's one way. Those are called blunt cutters because you wind up with a blunt end. The other way to do it is with what's called a sticky cutter, or sticky ends. And these, they will cut at a specific part of the sequence they recognize. So if we look back at the very first sequence I have up there, the ATGCAT, the way most cutters work is they will, if they bind that sequence, they will cut between the first A and the first T. Okay. Now, if you look at the other strand of DNA, the first A and the first T of that strand are on the opposite side. It's the 5' most end of the strand. On each strand, it's 5', which means that you're cutting the very first base off, and the last 5 bases are on the other side of the cut. And on the opposite strand, the first base and the rest of the 5 bases, but that winds up leaving you with 4 bases that do not have a base pair anymore, because the DNA has now denatured at that cut site. And I think the next slide shows that better. So if you look at this, this is the red part of the image at the top center. That is the restriction recognition site of a restriction enzyme called BAMH1. And if you look at the dotted red line in between the bases, that is how it cuts. So the brown arrowheads point out where on each strand of DNA the phosphate backbone gets cut or cleaved. And when those are cut, the two sides of double helix sort of move apart from each other, and now you have these sticky ends of 4 bases hanging off. That's the part in red at the bottom of the image, on the left and the right. These are sticky ends. Now, we can anneal that. If we have another double helix of DNA with the 4 matching bases hanging off the end, then they can anneal to each other. Now, I am not telling you that it has to be the other side of the same double helix. I am telling you it can be any double helix that has the correct 4 bases hanging off of its end on the correct strand. So, for instance, if I have a circular bacterial DNA chromosome, and I know that it has a BAMH1 site in it, and I have a gene from a human DNA that I'm interested in, and I find that it has a BAMH1 site on either end of that gene, I can, in one test tube, cut the bacterial chromosome, and if it cuts at that BAMH1, it will now be an open chromosome with 2 BAMH1 sticky ends. And then in a different test tube, I cut that human gene with BAMH1, and now each of the 2 BAMH1 sites, on the one on either side of it, those are cut, and so it has 2 sticky ends on it. And if I put that in the same test tube as the cut bacterial chromosome with the 2 sticky ends, and then I let them anneal, in a lot of cases the bacteria is going to anneal to itself, or the gene is going to anneal to itself, but in some cases it's going to anneal closed with the human gene inserted into the bacterial chromosome. This is what molecular biologists refer to as cloning. This is not the popular science, popular media version of cloning, where you make an identical genetic copy of a different, live animal. This is not Dolly the sheep, which I'm realizing now probably predates all of your births. This is the standard molecular biology definition of cloning, and that is moving DNA around and inserting it where it doesn't belong, but where it will be helpful to you. That is going to be part of next week's lab also. Not this week's lab, though. But, so, I was going on in that aside about Rich Roberts, about how scientists were so excited about restriction enzymes, because they could do so many wonderful things. Well, I didn't say what the wonderful things they could do were. So let's talk about that. One of the things is you can do cloning. This is incredibly helpful if you want a particular gene to be expressed in a particular place and time in an organism. You can take the gene and clone it so that it matches up with the sequences of DNA that control where and when it's expressed, called an enhancer. And you can combine that with a sequence of DNA called a promoter, which is what causes it to be turned into RNA from DNA. And you can control all of these things. You can take the promoter from this gene and the sequence of that gene and the enhancer of that gene and mix and match and combine them all. There's many other parts of this process that I have not taught you yet, that you probably have not been exposed to yet, that can also be moved around and mixed and matched. And huge quantities of today's medical and biological research just involve this. This is a huge part of molecular biology laboratories. There's another technique that's used called restriction mapping. And it's not as common now, but it is still used. I'll tell you that my first undergraduate lab job way back in the mid-1990s was performing restriction mapping. The lab I was working in, multiple other people, including myself, we created a restriction map that spanned 4 million bases of DNA on a human chromosome. Now, what do I mean when I say a restriction map? What I mean is you take a chunk of DNA. In this case, on this slide, you're looking at a 10 kb circular DNA chromosome. And you take that and you digest it with a restriction enzyme. And in this case, the enzyme EcoR1. And when you digest that, the enzyme cuts up the DNA, and then you run that on a gel, and you see what size the fragments of DNA are. You know that they're going to be a total of 10 kb added up. In this case, you get one band of DNA that's 6 kb and one band of DNA that's 4 kb. Okay, great. What does that tell you? Well, it tells you that in your circular DNA, there are 2 EcoR1 sites, and they are, in one direction, 4 kb apart from each other, and in the other direction, they're 6 kb apart from each other. I hope it is clear to you how we figured that out. The fact that when you cut the bacteria with this enzyme, and it cuts at specific DNA sequences, we know how far apart those DNA sequences are by how large the fragments of DNA are left over afterwards. Okay, great. So we know that. Big deal. That doesn't really tell us anything. We don't know where any of the genes are. We don't know why there's EcoR1 sites. We don't know any of the DNA sequence. We just know that somewhere in this circular plasmid, or circular chromosome that's 10 kb long, there are 2 EcoR1 binding sites, or restriction sites. Well, there's literally hundreds, possibly thousands of restriction enzymes that are known to modern medical science. So let's pick another one. We're looking at the same DNA, and now we're picking an enzyme called BAMH1. And that's the one that we looked at 2 slides back. Now, the BAMH1 also cuts this plasmid DNA twice, but the 2 BAMH1 sites are not at 4 kb and 6 kb apart from each other. They are 3 kb apart from each other in one direction and 7 kb in the other direction. Okay, so it has 2 BAMH1 sites, and they're at different distances apart from each other than the EcoR1 sites are. So what? Well, where are they? Where are the BAMH1 sites in relation to the EcoR1 sites? I don't know. Do you know? No. In both of these past two slides, we have shown one of the cut sites at the 12 o'clock position of the circle, and the other cut site somewhere between 6 and 9 o'clock on the circle. The EcoR1 site was somewhere around 7 o'clock, let's say, and the second BAMH1 site is at about 8 o'clock. But that doesn't mean that that's where they are in relation to each other. I mean, that's where they are in relation to, BAMH1 to BAMH1 or EcoR1 to EcoR1. But we have no idea that the EcoR1 site that we drew at 12 o'clock is in the same place as the BAMH1 we drew at 12 o'clock. They could be anywhere with respect to each other. So let's find out where they are. They could be anywhere. The BAMH1, the one that we drew at 12 o'clock, could be at 9, it could be at 3, it could be at 2. Who knows? So what we do then is we digest with both enzymes at the same time. And we look to see what size bands of DNA we get at that point. Now, a couple of things to keep in mind. The number of fragments you get tells you the number of cut sites you have. But there are two possible options. Whether you start off with circular DNA or if you start off with linear DNA. How does that make a difference? Well, think of a rubber band. Okay, we've got a rubber band, that's circular, and we've got a pair of scissors. What happens if we cut that rubber band twice? Well, the first time we cut it, it goes from a rubber band to a rubber strip. The second time we cut it, it goes from a rubber strip to two rubber strips. So starting with something circular and cutting it twice, we end up with two fragments. But what happens if we don't start with a regular rubber band? We start with a rubber band that's already been broken. It's already a rubber strip. What happens when we cut that strip twice? The first time we cut it, we wind up with two strips. Now we're going to cut one more time, pick either one of the strips, and cut that. Now we don't have two strips, we have three. So if you cut a circular DNA, the number of times you cut equals the number of fragments. If you cut linear DNA, you have one more fragment than the number of cuts. That is important to understanding how to interpret restriction digests and how to generate restriction maps. Now, the sizes of the bands of the double digest tell you, well, first off, flat out, the sizes of the bands of any restriction digest tell you how far apart the restriction sites are that cut that band, that fragment of DNA. If the fragment of DNA is 4 kilobases long, then it had one restriction enzyme at one side, and 4 kilobases away on the other side, it had the other restriction enzyme site. That's the same whether you have one enzyme or two enzymes or 20 enzymes. One end of it got cut, and the other end of it got cut. And the length of the fragment of DNA is the distance between the two places it was cut, whether it was cut by the same enzyme on both sides or different enzymes on both sides, or different enzymes on each side. So let's look at this. From our EcoR1, we have a 6 kb band and a 4 kb band. And from the BAMH1, we have a 7 kb and a 3 kb. And in the Eco and the BAM together, we have 1, 2, 3, and 4. Now the first thing that you might notice is you have a 3 in the BAM and a 3 in the Eco and BAM together. Maybe that's the same stretch of DNA. Maybe. But let's think this through a little bit more. Look at the 7 kb band in the BAMH1 site. Where did that go? Well, it's possible that where did it go? Where it went is, it got cut. It got cut into smaller pieces. How? Well, it must mean that somewhere inside that 7 kb band, there is an EcoR1 site. Maybe one, maybe two. We don't know. So it could be that there is one in there that is 4 kb from one side and 3 kb from the other. Or it could be that there's two in there, and one of them is 1 kb apart. Two of them are 1 kb apart, and then two are 2 kb apart, and then there's one that's 4 kb apart. So you have to think through what it might mean, how it could happen. Well, what happened in the EcoR1 side? What happened to the 6 kb band? Well, that got cut, because it's not there anymore. So maybe that's a 3 and a 2 and a 1, or maybe that's a 4 and a 2. So what you wind up with here is a logic puzzle. How do I get from 6 and 4 and 7 and 3 to 4, 3, 2, 1? There's a couple of ways you can do it, but most likely there's only going to be one way that you can do it that keeps the E. coli sites 4 and 6 kb apart from each other, and the BAMH1 sites 3 and 7 kb apart from each other. And remember, you only have 2 EcoR1 sites and you only have 2 BAMH1 sites. And so you have to figure out where on the circular DNA chromosome they are in respect to each other to find out the answer. So you have to start spinning the circles of DNA with respect to each other to see how you get to an answer. And in this particular case, we get the 1, 2, 3, 4 by placing our restriction recognition sites in this orientation to each other. So if we keep that EcoR1 site at the 12 o'clock position, then 2 kb away from it, going clockwise, we now have a BAMH1. And so when we cut this DNA with both EcoR1 and BAMH1, those two specific cut sites are going to generate a 2 kb band, and that's the 2 kb band we see. And then let's look at what other bands are generated. Moving clockwise still from that BAMH1 site, 4 kb away, we hit the EcoR1 site. Now, first thing we look at is, if we didn't have that BAMH1 site there, the 2 EcoR1 sites would be 2 plus 4 kilobases away from each other. That's 6 kb. That is what the EcoR1s were originally. So those are still on the same relative orientation, relative positions to each other as they started off. Great. And so on down the line through the whole thing. You can see that you generate all the correct fragments from this. Now, if you had the mirror image of that, that's just as valid, because we don't know what the open reading frames are, we don't know what strand has the gene on it, or what direction the bases are going. We just know how far apart the DNAs are. Now, what you can do with this is, there are artificial bacterial chromosomes called plasmids. There are also cosmids and a few other things. They mean slightly different things. But a plasmid is an artificial bacterial chromosome made by humans in a lab that is somewhere between 2 and 12 kb long. It has an origin of replication so that it can get replicated during S phase. It has several restriction sites in it, usually in a specific small region called a multi-cloning site, and there's more information on that in the lab handout for this week. And it usually has an antibiotic resistance gene, so that when you try and grow this in lab, you can add an antibiotic and it'll kill every bacteria that does not have your plasmid inside it. And we can take that and we can insert whatever DNA we want up to about 12 kb. And in the lab I worked in, for example, we had, or people before me in the lab, before I joined, had generated thousands of plasmids that had chunks of DNA in them, chunks of human DNA, from human chromosome 22q11. And there were thousands and thousands of plasmids, and most of them had, redundant sequences with each other, and, so more than one plasmid had the same DNA sequence, or plasmids had DNA sequences that overlapped with each other, and so on. And what we did was, we would systematically, one by one, digest these plasmids with multiple different restriction enzymes and find out what the pattern of restriction sites was in each plasmid. And from that, we could figure out, we knew right away which part was bacterial and which part was the human DNA, because we had the bacterial information already determined. But we took the human restriction maps that we had generated of just the stretches of human DNA we had put into these plasmids, and then we started piecing them together and saying, okay, this plasmid has a stretch of human DNA that has these 4 restriction enzyme sites located the same distance from each other as that chunk of human DNA. So they probably overlap with each other. And so then we would say this plasmid overlaps that plasmid. And then we'd find another plasmid that overlapped that one, and another one that was redundant with this one. And then we would be able to line them up like dominoes, not standing dominoes, but like the game dominoes where you match the 2 and the 2 and the 6 and the 6 and the 5 and the 5. We would match up the restriction sites of these different chunks of human DNA, 4 to 10 kb at a time, That's 4 to 10,000 bases at a time for 4 million bases. And once we had that, we knew, okay, here's a stretch of human DNA 4 million bases long on this part of this chromosome. Somewhere in this region is a mutation, or somewhere in this region, if you get a specific mutation, you wind up with a disease called DiGeorge syndrome. It's a genetic disease. And at the time, no one knew what caused it. They knew roughly where on chromosome 21 it was, or I'm sorry, chromosome 22, where it was, but they didn't know what gene it was. They didn't know what sequence it was. They didn't know what the mutation was. It was known that a lot of people who had the disease had a deletion somewhere in there. So what did we do? We started comparing the DNA of patients with this long map that we had generated and said, okay, the patients all have different deletions, but they all have the same region deleted, and that region corresponds to this fragment between these restriction enzyme sites. Great. This was at a time when sequencing was expensive, hard, and slow. Creating this restriction map over the course of 2 years was much faster than trying to sequence that entire section of chromosome 22. Now you can sequence an entire genome in an afternoon for $1,000. Back then, it would have taken over $1 million and over almost a year. So this was the cheaper, easier, but still slightly slower solution. And based on that restriction map, we were able to find out that the mutation or deletion that caused DiGeorge syndrome was in a gene called TBX1. Okay, so I spent a lot more time on that than I had planned. So for this, this starts with a linear DNA, and I want you, as your homework for this lecture, to create a restriction map on the linear DNA of where the EcoR1 and the HinD3 sites are and how many of them there are. You already have all the information that you need between the previous couple of slides of explanation and the information on this slide. You can solve this answer. You can solve this problem. And there are two possible solutions. One reading left to right and one reading right to left. You can get the mirror image solution. Either one will be correct. I want to move on now. These next two items are two of the biggest discoveries of biological techniques in the history of molecular biology. Now, granted, that's a field that's only been around for 100 years, but still, these are the two biggest events. Both of them yielded Nobel Prizes. So we're going to start with polymerase chain reaction, usually referred to just as PCR, and then we'll go to DNA sequencing. Specifically, we're going to go into Sanger sequencing, but there are two types of DNA sequencing. So, what is polymerase chain reaction, or PCR? It is, quite literally, a chain reaction using DNA polymerase. So it is a well-named reaction. It was invented at one of the first biotech companies in 1983, the Cetus Corporation, by a scientist named Kary Mullins, and it was improved upon by one of his co-workers, Randall Saiki. So what is it? Well, strictly speaking, polymerase chain reaction is a technique to make billions of copies of a small stretch of DNA in a small period of time. I have included here a link to a YouTube video. There are a lot of videos on PCR. I didn't like most of them, honestly. This one is decent. It's not the best. I think I could probably make a better one, but it's the best of what I could find to help explain the process. I'm going to explain the process here, and then after you've watched the rest of this lecture, go watch the YouTube video if you don't understand. So what happens is basically you set up the DNA polymerase replication reaction in a test tube with specific DNA that you want. Now, if you remember the previous lecture, replication requires a lot of different proteins, requires DNA and RNA, nucleotides. It's a whole huge process, clamps and loaders. This is a simplified version, and I can explain to you how. I talked a lot about denaturing and annealing at the beginning of this lecture. Well, I also mentioned that DNA helicase breaks up the two strands of the double helix into single-stranded DNA. That's the same thing that denaturing does. So if we can denature the DNA in a test tube, we don't need helicase. So we can skip including helicase and all the clamps and loaders required for helicase. And we can also skip topoisomerase because the vast majority of the time we're working with linear DNA, and even when we're working with circular DNA, we don't care if it gets supercoiled. We just care if we can access the region of the DNA that we need. And that's all. So we don't have to worry about topoisomerase, helicase, clamps, or loaders. Already, we've cut down significantly on the amount that we need to insert in here. We're also doing this in a test tube, not in a live cell, so we don't need single-stranded binding protein because we don't have the negative environment that will destroy the DNA, and we also don't have the cellular proteins that seek out single-cell DNA and destroy it thinking that it's viral. So we can get rid of the single-stranded binding protein. And if you remember, I mentioned in this slide in the replication lecture that there are two types of DNA polymerase 1, or generally two types of polymerases, processive and non-processive. And I talked about how the non-processive polymerase can just add one nucleotide before it falls off, and that you need the DNA clamp to hold that polymerase on the double helix or the single helix region and push it forward so that it can add more than just one base. That's the non-processive. But there's also processive polymerases, and they can add thousands of bases one after another before they fall off the DNA and dissociate from it. So in PCR, we're going to use a processive DNA polymerase instead of a non-processive one. The other thing that we can do, as I said, processive, not the non-processive. The other thing that we're going to do is, instead of needing an RNA primase to generate random small RNA primers to allow the replication to occur, we are going to include in this test tube specific primers that we want, not random ones, and we're going to make them out of DNA, not out of RNA. Why? Well, if we make them out of DNA instead of RNA, we don't need the polymerase 1 with its 5' to 3' exonuclease activity to come in and chop up the RNA and add DNA in its place. We already have DNA there. So we don't need two different polymerases, we just need the one. And we are making specific primers because we want replication to start at a specific place. So really, all you need for PCR is you need your DNA and you need nucleotides to generate new DNA. You need your primers and you need polymerase. And the polymerase itself needs magnesium to function. I believe I mentioned that in the previous lecture, that the magnesium sits in the palm of the palm-shaped polymerase and it helps bend the nucleotides in the right orientation to catalyze the reaction. So you need that magnesium or else it won't work. So how does PCR work? And this is beautiful. Honestly, Kary Mullis was a surfer, an inveterate surfer. Cetus Corporation was located on the coast, and he would go out every day after work and surf all night long. And he was kind of a stereotypical California airhead surfer. If that's a stereotype that you guys still know, it was popular when I was younger. He was trying to work with a region of DNA, and he needed multiple copies of it, and it took forever to grow more bacteria, purify the bacteria, digest its DNA, cut it up, and then have enough to do what he needed to do. It was expensive and it was slow, and he was trying to come up with a faster way. And he said, if only we could do replication in a test tube. Well, what would you need to do replication in a test tube? And then he figured it out, and boom, lo and behold, you can do it pretty easily with just heat and polymerase. So you put all these things that I listed into a test tube, and you heat it up to denature the double-stranded DNA. Okay? And then as it's cooling to anneal, just like before with that 1,000-base DNA primer on the chromosome, in this case, you've got a 20-base primer on a region of DNA that, who knows how big it can be or how small. But the 20 bases is going to bind to its perfect match sequence on the larger DNA faster than the other strand of the full DNA double helix will bind. And so then you have your primer bound to the single strand of DNA that it needs to bind to, and the polymerase can come along, grab that, and if it's at the right temperature, and if it has the magnesium and the nucleotides, and a bunch of ATP, it can start polymerizing DNA. And it makes a second copy of that strand of DNA. Okay, great. Now what? Now you heat up the whole process, you heat up the entire test tube again, and all of that DNA denatures, and then you cool it off, and the primer anneals to the DNA again, and then it replicates again, and you keep doing this process over and over and over again. If you do these 3 steps, denature, anneal, and elongate or replicate, if you do that once, you have doubled your amount of DNA. If you do it twice, you go from 2 copies to 4. The third time, you go from 4 copies to 8. So we're looking at powers of 2 here. 2 to the first, 2 squared, 2 cubed. If you repeat this 3-step process 30 times, you have 2 to the 30 copies of DNA. Now, if I remind you that polymerase moves along at a speed of about 1,000 bases a second, you can replicate large quantities of DNA very quickly. In the lab, we generally use the mindset of you want 1,000 bases per minute instead of 1,000 per second. That's what we use experimentally. But under ideal conditions, it can go that fast. So each one of these 3-step cycles can be as short as 45 seconds or as long as 3 or 4 minutes. So doing 30 of them at most is going to take you 3 or 4 hours. It could be done in as short as an hour 20. There's a few other steps on either end to help make everything neat and tidy, but this is the core of the process. Now, when Kary Mullis first invented this, he was using a DNA polymerase that he had isolated from E. coli. And the problem was, when you heated up the test tube enough to denature the DNA, it also denatured the protein. Because, lo and behold, proteins are one long polymer of amino acids covalently bound to each other, and then that polymer bends and folds on itself and is held together by hydrogen bonds. So the shape of a protein is most often held in place by hydrogen bonds. And when you heat up the test tube enough to get rid of the hydrogen bonds in the DNA, you're also getting rid of the hydrogen bonds in the protein. And when you cool it off to anneal the DNA, apparently proteins cannot reform in the correct shape just by cooling enough to reform hydrogen bonds. It's just like cooling DNA too fast, the proteins just can't form the right shape. They're a mess. And so the polymerase, every time you went through a cycle, once you heated it up to denature the DNA, the polymerase was inactive. So you had to add new polymerase every single cycle. And that got to be very time consuming and also very expensive, because it was expensive to purify polymerase. And what Kary Mullis' co-worker, Randall Psyche, did, his innovation to the process of PCR that made it so much faster and so much cheaper, is he looked at a species of bacteria called Thermus aquaticus. Thermus aquaticus lives and thrives in hot springs, where the temperature of the water is almost 94 degrees, the temperature that you're denaturing the DNA at. And if the bacteria can survive at that temperature, then there's no way that the proteins are denatured at that temperature. They've got to be stable somehow. And so he purified the protein, the polymerase protein from Thermus aquaticus, and he tried that in PCR, and he did not have to add new polymerase every single cycle. The one polymerase worked throughout the denature and reanneal cycles, so it was not getting denatured at the high temperature. So people wound up cloning the gene that made this protein in Thermus aquaticus, and then expressing that in E. coli and all sorts of other bacteria so that they could purify large quantities of it without having to go to thermal vents and hot springs to get it. And so now that protein is called Taq polymerase. TAQ, the T for Thermus and the AQ for aquaticus. So Taq polymerase is one of the most commonly used enzymes in molecular biology because it is required for polymerase chain reaction. Oh, and by the way, these guys got the Nobel Prize for their work. So let's look at this process, shall we? So this is a figure from the book, and I actually, looking at this static figure, I don't feel like it explains the process very well. But it does explain the process. So at the top you have your double-stranded DNA with the strands going in opposite directions. You heat denature so that those two strands separate. And then the little green arrows are your primers. And they bind to your DNA of interest. And then you undergo the process of replication. And so now I'm at the third row of this image. There was a double helix at the top of the page, and then the denaturing and the binding of primers at the next row, and then the third row is you've now got two copies of the double helix. And the sort of faint red lines are the newly synthesized replicated other helices. Okay, so now in your test tube you have two double helices of the exact same DNA sequence. And you go through the process again, and you denature and primer anneals, and then you go through replication. It's called elongation in this case because you're elongating the sequence of DNA. And now at the end of that, you have 4 copies of your double helix. And then you go through that again, and you get more and more and more and more copies. I made my own slide to show this in a little bit more detail. Because you're not just amplifying all of your DNA. The way PCR works in real life is you've got a big chunk of DNA and you want to amplify a small part of it. And you don't care about the rest of it. You just want your specific region. So in this case, we have a double-stranded, a double helix of DNA, and that's the lines in black. And the region of DNA that we are interested in copying, I have highlighted in red. So it's just the middle third. So we create primers that match the sequences on either end. And the primers have to run from the 5' to 3' direction. Okay, so we put all of this in our test tube with our Taq and our magnesium and some nucleotides, and we heat denature, and then we allow the primers to bind. And they do. And then we put it at the temperature that the polymerase works best at, and the polymerase starts off at our primers and just proceeds until the end of the DNA. So we have created 2 double helices of the region of interest to us, but we also have some other DNA that's been replicated also. Okay, that's not a big deal. I mean, we don't need it, but it's not hurting anybody that it's there. But let's do the process again. We anneal our primers, we denature the DNA, then we let our primers anneal, and then we replicate again. And now, as you can see, we have some DNA that is just the region that we're interested in amplifying. And it has none of the non-red DNA. I mean, there's still some that's double helix of the black DNA or single strand of the black DNA, but we've now generated some of the DNA that we want. And we will repeat the process again, and that time we'll end up with a lot more of just the part of the DNA we want. And after several cycles, the sample becomes overwhelmed with just the DNA you're interested in. And so if you've got 10, 15, 32 copies of all of that DNA labeled in black, but you have 2 to the 30th copies of the DNA labeled in red, then you're not going to worry about the 10 to 15 copies when you've got billions and trillions of the right size and the right DNA. So that is PCR. And you can do all kinds of crazy things, because PCR, you're not just amplifying your DNA. I mean, you need that for other things, and you can cut it with restriction enzymes, you can put it in other solutions, you can do all kinds of things with it. But you can also make funky primers. And in this case, what I mean by that is, if you have a primer that is 20 bases long and it matches your DNA perfectly, that's great. That's gonna hydrogen bond the heck out of the double helix of DNA. But then, let's say on the 5' end of that, you add a handful of bases that don't match. Well, you've got 20 bases of match. The hydrogen bonding of those 20 bases is going to be pretty strong enough that the bases that you're including that don't match at the 5' end aren't going to be a big problem. So you put there specific bases that you want that do specific things. For instance, you could put specific bases that match the restriction site of a restriction enzyme, so that when you run your PCR, after 2 cycles, if you look at this and draw it out on a piece of paper of your own, what you realize is after 2 or 3 cycles, the DNA that you're amplifying, that red part, includes the sequence of DNA on the 5' end of your primers that didn't originally match. But now you are amplifying that part, and so it is matching the stuff that you're generating. And so at the end of your 30 cycles, you have billions of copies of this red region of DNA plus those extra bases you added on the end of your primer. And then you can take that and you can add restriction enzymes, and they will cut at the ends the parts that you inserted on your primers. And now you have your region of DNA that you were interested in, and it has sticky ends from the restriction sites. And you can use those sticky ends to anneal it into a plasmid of your interest that you can then use to do other things with the gene. This is cloning, and this is how people make fluorescent cells. This is how people make it so that you can follow a particular RNA being expressed, or a particular protein floating around in a cell. You can make that protein fluorescent. So the whole cell isn't fluorescent, just the parts where that protein is. This is how you can make certain membranes in a cell, or certain pores in a membrane, fluorescent. You do all of this. Or you can make it so that that a protein that has to be in the nucleus is trapped against the cell membrane and can't get into the nucleus until you add a certain chemical like estrogen or ampicillin. And then adding that chemical breaks the thing trapping it against the cell membrane, and then it can go into the nucleus and do its job. So when you do that, you can make it so that a particular protein does not function until you want it to. And you can see, oh, look, the mouse that I've done this to is perfectly happy and fine. Now, let's add in some of this, ampicillin or penicillin or estrogen, and that's going to activate the protein that we had deactivated, and now let's see what happens. And you go into this with a hypothesis of what you think will happen, and you see if you're right or wrong. So these are just a few of the millions of aspects of PCR that are incredibly helpful. Another one is, let's say you're at a crime scene, and your perpetrator left behind 2 hairs of their own with roots, or a little bit of a semen sample, or blood. And you can get some DNA out of that, but not a lot. Well, if you want to do short tandem repeat analysis to do DNA fingerprinting, you can perform PCR across those tandem repeats to see if they have the right number of repeats to match your suspect. So you can amplify small quantities of DNA found at a crime scene to make enough DNA to perform criminal analysis and DNA fingerprinting. So, we are at the end of our time for this lecture. I only have 5 or 6 slides on DNA sequencing, but I'm going to save those for the next lecture. So we will stop here for today.